Yeah, not so quiz is on Thursday. Yes, whatever we have finished up to today will be in the quiz.
Thank you.
Yeah. Okay. So last time we did the generation and transmission of action potential, and today we're going to talk about the some other section of the chapter 7. This is refractory period. So once you stimulate a part of the axons in the neuron, during a certain period of time, you cannot re-stimulate it. And that small period of time is called absolute refractory period. Okay. So let's see, this is the timeline. This is the voltage. So let's see, if you stimulate the plasma membrane of neuron here, what happens? Once it reaches the threshold, which is 50, then it gives the action potential, depolarization, repolarization, hyperpolarization. So this is one action potential. So up to the peak of this potential is due to the opening of sodium channel. So when sodium channel is open, that means every micro inch of the axon is depolarized. And during that small period of time, the channels, once they are depolarizing, once it depolarizes for that small period of time, the channel is inactivated by the polypeptide ball and chain channel. Do you remember there was ball and channel when activated, they close the channel. So immediately another activation cannot stimulate it. Okay. So absolute refractory period is due to inactivated sodium channel, and that sodium channels are, you can see in the note, inactivated by ball and chain polypeptide channel, and that is refractory period. So you see, refractory period is around about 0 to around 1 millisecond. So if you, let's say, you stimulate a neuron and then you give another stimulus within 1 millisecond, then you will not produce this action potential because the sodium channels are now temporarily blocked by ball and chain peptide. Okay. Now, once hyperpolarization here, once repolarization, then hyperpolarization start, you can activate the axons with a stronger stimulus, and that is called refractory period, which is relative refractory period. And this is due to continued outward diffusion of potassium. So in hyperpolarization, as you know, there is continued potassium going outside. That means if you stimulate, you can open the sodium channel, and then sodium can get inside and bring the another stimulus in this, but you need stronger stimulus. So refractory period occurs when voltage gated potassium channel are open, making it harder to depolarize the threshold. Okay. So absolute refractory period is due to inactivation of sodium channels, and relative refractory period is due to continued outward diffusion of potassium. During that time, in absolute, you cannot at all, but in relative, if the stimulus is stronger, you can open it and give another stimulus. Okay. Now, once action potential is produced in the axons, then how it conduct from every micro inch to other micro inch. Basically, the length, whole length of action, like every micro inch has to, action potential has to pass. Okay. So that is called axonal conduction. Here you go. So axons are two types, you know that. Axons are either myelinated or unmyelinated. If Swann sheath is covering the myelin, then we call them myelinated axons. If they are not covered, then we call them unmyelinated axons. Uh, so the speed of action potential in unmyelinated axons and the speed of action potential in myelinated axons are different. There is slower speed in unmyelinated axons and faster speed in myelinated axons. Okay, so why is that? Let's see here. So let's see in this neuron here, once you give a stimulus to dendrites, because dendrites are outside, so you can see dendrites, when there is any stimulus like touch, pain, pressure, heat, anything, when you give stimulus here, there is change in plasma membrane permeability, and that all added up and comes to the axon healer. And in the axon healers, there is a lot of sodium channel. Sodium channel open, and then sodium get inside, potassium come out. And then every microinch slowly, the sodium is in, out, in, out, in, out, in, out, sodium and potassium, and that becomes the current, electricity. And then it comes to the end of the axon. That end of the axon is called axon terminal. That release neurotransmitter and communicate with another neuron. So what happens here, you see here in this section of the axon potential, let's a box, section of axon here. And we have denoted resting potential where there is no stimulus is the amber color, depolarization blue color, and repolarization slightly pinkish color. So you see here, number one, when you give a stimulus, this is the resting membrane potential. When you give a stimulus, what happens? You open the sodium channel, and that is called voltage gated sodium channel. So when voltage is due to a stimulus, when voltage gets opened, sodium get inside, and then what happens? Inside was earlier negative, now it becomes positive, but here in the resting is still negative. Now another section of the axon opens. So this sodium channel opens sodium channel here now. As you remember, opening of sodium channel is positive feedback mechanism. So opening here, more sodium in, then this area opens sodium channel here, and that's how sodium is. Like you have negative, positive, negative, positive, negative, positive, in, out, and that's how electric current is passing in the actions. That means the ions have to flow in every micro inch, micron inch of, or micron centimeter of axons. That's why axon potential AP is slower in unmyelinated actions. You will see next time in myelinated. In myelinated, the action potential is faster. Speed of action potential is faster. Okay. So generating a new AP action potential process repeats all along the axons. And so action potential amplitude is always the same. Okay. And conduction is slow. Let's see myelinated action. So this is the section of action here now, but it is myelin. You see myelin sheath here, 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 and there is gap between the myelin sheath. What is that gap called?
Node of Ranvier.
Node of Ranvier. Good. That is called node of Ranvier. So myelin is fat. That means this is bad conductor of ions flow. So wherever myelin is covering, sodium and potassium cannot get in and out. So what happens? Once you give a stimulus to the resting membrane here, they produce action potential in the gap, and then the ions then just flow from here all the way to the gap. And then again, sodium and potassium get in and out, and then sodium here. So basically, ions are like jumping. It feels like they are not jumping, they are going inside, but it feels like it jumps from here to here. This section does not need to allow to flow the sodium and potassium. So it is jumping. That's why conduction in myelinated axon is faster. Ions cannot flow across myelinated membrane. Action potential are produced only at the nodes of Ranvier. Thus, no action potential occur under myelin and no current leaks. This increases the current spread and increase the speed of action potential. Okay. This kind of action potential is called saltatory conduction. Okay. They are fast because action potential script from node to node. Okay. Now, synapse. So what is synapse? We have millions of neurons in our body. Neurons have cell body, dendrites, and axons. Both dendrites and axons communicate with either other dendrites, other axons, or other cells in the body. But they are not contacting with each other directly, like my finger is touching with each other. No, neurons contact through a gap between them. And that gap is called, what is that gap called?
Synaptic
Synapse.
cleft.
Synaptic
Synapse.
cleft.
That gap is called synapse. Synapse, and they are not anatomical junction. They are functional junction. They are non-anatomical junction because they have not physical contact with each other. So that gap is filled with a chemical substance that is called neurotransmitter, and that neurotransmitter carry impulse from one nerve ending to another nerve ending or to another cell. Are you following me? So here, synapse is non-anatomical junction, which is gap between an axon and another neuron or target cell. Okay? And this synapse are two types. When two cells communicate with each other by a chemical compound in between them, then they are called chemical synapse. If they communicate through the ions, flow of ions between them, then we call them electrical synapse. Okay, so let's see here. Synapse is a functional non-anatomical connection between a neuron and neuron. Before the synapse is called presynaptic neuron, and another cell which is postsynaptic. If that is postsynaptic cell, then that will be postsynaptic cell. If it is a neuron, then that will be postsynaptic neuron. So let me show you. I'm going to go all the way. So here is two set of neuron. You see here, this is the axons of first neuron. That's why you call them presynaptic axons. And this is postsynaptic neuron. Are you following me? So presynaptic neuron is attaching to the cell body. Sometimes they attach to the dendrites. You see here? But it looks like they are attached anatomical, but they are not. If you see with electron microscope, you're going to see there is a gap between them. And that is called synapse. And the neuron before this is presynaptic neuron, and this is postsynaptic neuron. Okay, so let's go back. Okay, there are chemical and electrical synapse. Synaptic transmission at a chemical synapse is via neurotransmitter, as I said earlier. But electrical synapses are rare in the nervous system, and they are found in cardiac and smooth muscle cells. So let's see here, start with the electrical synapse. So here we have two cells. As I said, electrical synapse is common in cardiac muscle, smooth muscle, some glial cells, and some neurons. Very rare in neurons. Okay, so let's see, this is a two cardiac muscle cells and their plasma membrane. This is the plasma membrane of one cell here, and here is plasma membrane of another cell. These two cells are connected to each other by a whorl of protein called connexin protein. These connexin proteins are gap junction. Do you remember if there are two cells, epithelial cells together, or any other cells together attached to each other, they have a tube made up of connexin protein, transmembrane protein connection tube, and those transmembrane proteins sometimes act as junction, and that is called gap junction, and those gap junctions are electrical synapse. This gap junction in cardiac muscle cells are electrical synapse. And what is the another name for that gap junction in cardiac muscles? People who are taking human anatomy can answer that question.
Intercalated discs.
Intercalated disc. Good. That is called intercalated disc, or we can call them gap junction in other cases. Okay. So what happens in this case? Let's see this. If we stimulate this cell and open sodium, and then sodium get inside this cell, then what happens? Sodium can easily pass through this channel and go to cytoplasm of another cell. Okay. So depolarization, which is sodium flow or any potassium ion flow, depolarization flows from presynaptic into postsynaptic cell through the channels that is called gap junction. And these gap junctions are formed by connection proteins. Okay. We're not going to go in detail about that. So now, chemical synapse. Chemical synapse is usually between the neurons and other cells in the body. So if between the neuron, then we call them synapse or neuronal synapse. If they are neuron and muscle, between the muscle, then what do we call them? We call them neuromuscular junction. Okay. Let's see, presynaptic neuron instead of postsynaptic neuron. If this is muscle cell, then what do we call them? Neuromuscular junction. Neuromuscular junction. Our every skeletal muscle cell in the body, or every other muscle cells in the body, has this junction with the neuron. Otherwise, your muscle will not contract. It will not function. Okay. So here, this is the presynaptic neuron axon terminal, and this is postsynaptic cell, maybe neuron or other cell. So how chemical synapse work? Here we have number 1, 2, 3, 4. I want you to read it. Hey guys, when there is live class and when I see a student are sleeping, not active, I can pinpoint and say, hey, I ask them question and they get awake. And in this virtual class, I don't know who is sleeping, who is paying attention, or you just turn it on and you are doing other stuff. This is adult class, and I hope everybody pay attention, because you're going to miss a lot if you don't pay attention. It will be harder for you to go and only read and understand everything. So hopefully everybody is paying attention. Okay. So go ahead, read it. So if I follow back from this axon, where I'm going to reach ultimately to the axon and ultimately to the cell body. Yes. In the cell body of presynaptic neuron, there is raw endoplasmic reticulum, a smooth endoplasmic reticulum, there is ribosomes, and they produce the lipids, the proteins, and they are neurotransmitter. So those neurotransmitter produced in the cell body and then transported at the nerve terminal. See here. So when they transported by exoplasmic flow in the vesicles, you see these synaptic vesicles, the pink dots, those are the neurotransmitters. There are several neurotransmitters in the body. Neurotransmitter. Neurotransmitters are like epinephrine, norepinephrine, serotonin, dopamine, GABA, GABA, glycine, aspartate, nitric oxide, acetylcholine. There is hundreds. Okay. We're going to talk about that later. So let's see. When presynaptic neuron here is depolarized, the depolarization then axon potential from the cell body comes to the axons and then to the axon terminal. And axon terminal is already storing the synaptic vesicle full of neurotransmitter. So when axon potential reach axon terminal from the axons, then what happens? The axon potential is change in voltage, and that change in voltage here nearby nerve terminal opens voltage gated calcium channel. In the plasma membrane of the axon terminal, there is voltage gated calcium channel. So these voltage gated calcium channel, 1, 2, 3, 4, 5, they all open, you see. Calcium ions are more outside the cell. That's why calcium then rush in. Calcium ions move inside. And if positive ion is getting inside, then what happens? You further increase the concentration of calcium. Now this calcium is a kind of second messenger. So what it does, calcium binds to sensor protein in the cytoplasm. So in the cytoplasm of the axon terminal, there is another protein called sensor protein. And this calcium from outside come here and binds to the sensor protein. They make calcium protein complex. This complex brings these synaptic vesicles and stimulates fusion and exocytosis of neurotransmitter. You see, the vesicle is docking here and then they fuse with the axon terminal plasma membrane and then released by exocytosis. Now what happens? This red dot neurotransmitter, it may be acetylcholine. For these neurotransmitter, there is receptor on the plasma membrane of postsynaptic cell. They bind with those receptors. This is chemical, so they bind with chemically related receptor, which is called ligand receptor or chemical receptor, because these neurotransmitter ligand with the receptor, bind with the receptor. That's why we call these receptors here ligand receptors. Once they bind, what they do, they also open chemically gated channels in the postsynaptic plasma membrane. And again, extracellular sodium come inside the cell and depolarize this cell or this neuron. And again, axon potential is spread from there. That's how each neuron in your nervous system or in your body is working. Okay. So synaptic cleft separates terminal button from presynaptic nerve fibers from postsynaptic cells. So this is called terminal button, this whole thing. And this is synaptic vesicle here. This is synaptic cleft. Vesicle fuse with button membrane and release by exocytosis. So how much neurotransmission is released? It is based on how stronger a stimulus is. Stronger the stimulus, more the neurotransmitter release. And then once these neurotransmitter bind where, what happens? They bind for the on these postsynaptic forever? No, they are taken back, this presynaptic neuron, and they metabolize them, they reuse them. Okay, we're going to talk about that later. Okay, so now neurotransmitter release, axon potential reach the axon terminal, calcium enters. So this is all we have talked about. So this is just a language. Okay, so now once the produced in the cell body, released from the axon terminal, then there is reuptake and inactivation of the neurotransmitter. And we are taking here example of monoamine neurotransmitter, which is epinephrine, norepinephrine, dopamine. Those are the group of monoamine neurotransmitter. That's the example. You can take example of anything. Okay. So you can see here, as usual, up to now we know how it is released. So there is a release of the neurotransmitter. Let's see monoamine, how they make. In the cell body, monoamine, particularly the epinephrine and norepinephrine and dopamine, is made from an amino acid called tyrosine. So monoamine is, what are the example of monoamine? Monoamine is epinephrine, norepinephrine, and dopamine. So those monoamines produced from tyrosine and stored in synaptic vesicle here. So it's tyrosine, there is chemical process, enzymes, that enzymes turn tyrosine into dopamine, and from the dopa they form dopamine, and from the dopamine they are turned into norepinephrine, epinephrine, and they are in the vesicle. You see all the vesicle. As you know from the previous chapter slide, they are fused and then released. They're released, and once they're released, so what happens? Let's go again. Monoamine produced and stored in synaptic vesicles here. Action potential open calcium channels leading to release of neurotransmitter. We know that. Neurotransmitters enter synaptic cleft here. Once they release, they bind with the, hey guys, if your mic is turned on, please turn it off. Neurotransmitter, once entered the synaptic cleft, they bind with the ligand gated receptors. You see, these are the receptor on the postsynaptic cell membrane. So this purple neurotransmitter bind here. And once they bind, what they do, they open channels, sodium channels or other channels based on what kind of neurotransmitter they are. And then depolarize or hyperpolarize the postsynaptic cell. If they depolarize postsynaptic cell or neuron, that means postsynaptic cell is stimulated. If they hyperpolarize, that means postsynaptic cell or the postsynaptic neuron are inhibited. Okay. After that function, you see, slowly, once the function is done, once the channels are open, these neurotransmitters are released from these receptors and comes back in the synaptic cleft. And those neurotransmitters are then, by the active process using ATP, they are reuptaken of most neurotransmitter from synaptic cleft inside the presynaptic axon terminal. And then there are enzymes for a specific neurotransmitter, and that is inactivated. So inactivation of most neurotransmitter by, in this case, because this is monoamine oxidase enzymes. MAO is monoamine oxidase enzyme. Breaks down these all norepinephrine, epinephrine, and dopamine. Okay. Let's talk about some disease condition associated with it. So I'm going to talk about 3 disease or 4 or 5. Depression. It is in my note, you can see. Depression, anxiety, schizophrenia, OCD, which is obsessive compulsive disorder, ADHD. What is that? Attention deficit hyperactivity disorder. And then the most commonly used medicine in OCD, anxiety, depression is SSRI, selective serotonin reuptake inhibitor, which is called sertraline. Have you heard about this? Okay. So let's see, instead of this monoamine, the neurotransmitter here is serotonin. What does serotonin do? when you relax, when you exercise, when you hear the good news, when you feel valued. In that case, your nerve terminals release serotonin. And then serotonin bind with this receptor and relax the postsynaptic neuron. And you feel relaxed. You don't feel anxious or depressed. Okay? But if somebody has the neurotransmitter, which in this case, like in depressive case or the anxiety case or OCD case, the amount of serotonin is less here. Less means less binding, less relaxation of the postsynaptic neuron, and you feel anxiety. Are you following me? So to treat, what do we do? Just find serotonin somewhere and inject in your muscle or vein. Will that work? No, because that will not reach this synaptic cleft, because all the proteins and other things are metabolized by our liver, our blood enzymes. So what do we do? We may inhibit this step, number 4. We put the enzymes which can inhibit the reuptake of serotonin, and that is sertraline is. Sertraline inhibit the reuptake of serotonin, and whatever neurotransmitter is here works for longer time and relax you. That's why SSRI, sertraline, is the most common, safest medication for depression, OCD, and anxiety disorder. Okay?
Isn't there, Professor, isn't there always also a situation where the receptor is at fault? Like you can have a lot of serotonin, but then the receptor on the postsynaptic cell isn't
Yes,
receiving.
yes, that can be the condition too. But even in that case, if you increase the concentration, inhibit here, whatever neurotransmitter is there, that will be available for banding with the functional receptor for longer time, and that will improve the function of postsynaptic neuron. Okay, so in schizophrenia, what happens? This dopamine, particularly norepinephrine and dopamine, is a lot. So it binds a lot, and you feel all the time to go hyperactive. Your process of thinking is different. That happens in schizophrenia. In anxiety disorder, what happens? You have less. In the bipolar, in the bipolar case too, that norepinephrine is a lot. So these all mental disorder has biochemical association a lot nowadays, well understood. Okay, so this is the condition there. Now, presynaptic neuron and postsynaptic neuron. So let's see. This is presynaptic neuron, and here is postsynaptic neuron. If these presynaptic axons are depolarized and they are bringing the action potential here, so this one postsynaptic neuron may be synapsing with several presynaptic axons from several different neurons. Now, what will be the result in this postsynaptic neuron? Will this neuron will be inhibited or stimulated? This is based on what kind of neurotransmitter is released in presynaptic action here at in the synaptic cleft. Okay, so to understand that, we have to understand EPSP and IPSP. Okay, so let's see here, go back. Neurotransmitter diffuse across the clefts and bind to the receptor protein or postsynaptic membrane, opening chemically regulated ion channels. So when this presynaptic neuron depolarize the channel, that cause EPSP, excitatory postsynaptic potential. If they hyperpolarize, then they produce inhibitory postsynaptic potential. Okay. And these affect voltage gated channels in the postsynaptic cell. So let's see here. Let's see, this one is, there are 4 synapses. One is inhibitory because this releases the neurotransmitter which hyperpolarize this membrane. And 3 of them are depolarizing, which is stimulatory. So what will be the net result in this action?
Depolarizing?
Depolarizing. That's why this neuron will release the neurotransmitter, which is depolarizing, and activate this neuron. That's how presynaptic and postsynaptic neuron summate the action and brings it action. Okay. So synaptic, how the transmission is in synapse here, presynaptic neuron action potential conducted by axons, then opens voltage gated channel. And this summary: voltage gated calcium channel, release excitatory neurotransmitter. Then in postsynaptic neuron, dendrites and cell bodies opens chemically ligand gated channels. Inward diffusion of sodium causes depolarization. If there is inward diffusion of chloride, then that will be inhibitory. Localized decremental conduction of excitatory postsynaptic potential. And then action helic, again, voltage gated sodium and potassium channel, and conduction of action potential. This is how it goes all the way. Before I go to another slide, I want to remind you guys, local anesthetic lidocaine, which you get when you extract your teeth in the dental office by the dentist, they give you medication, lidocaine, and that numb your teeth because you don't get any depolarization, action potential. Why is that? What does that lidocaine do?
Blocks the
Blocks
sodium.
sodium channels.
Blocks the sodium channel. Good. So now we're going to go to chapter.
And then just to clarify, Professor, the dendrites and the cell body does not have any voltage gated sodium channels. They don't have that. Only the axon helic has that.
Yes,
Because
there are, but most are concentrated in that action helic.
that's because that's where action potential begins.
Yes.
On helic.
Yeah. Okay. So now central nervous system. In the central nervous system, what are the 2 parts of central nervous system?
Brain and spinal cord.
Brain and the spinal cord. So we're going to talk about a structural organization of the brain. In the brain, the uppermost part of the brain is called cerebrum. We're going to talk about that. Under the cerebrum, we have the diencephalon. In the diencephalon is in the middle of your brain and slightly lower part of your brain inside. And diencephalon has 3 parts: thalamus, hypothalamus, and epithalamus. We're going to talk about them. And then under the diencephalon, we have midbrain and hindbrain. We're going to talk about midbrain and hindbrain. And then after that, we're going to talk about in the midbrain, pons and medulla oblongata. And continuously then we will talk about spinal cord tracts and some cranial nerves and spinal nerves for the peripheral nervous system. So we will not go in detail of peripheral nervous system. We'll just talk about cranial and spinal nerves. That's why we skipped some of the part of the peripheral nervous system. Okay. So here, this is the CNS. CNS contains brain and spinal cord. So this is if you take my brain, you're going to see if you divide the brain into half, equal half, right and left. The diagram here is somebody's right side of the brain. So you are looking from the medial side of the right hemisphere of the brain. And you're going to see what are the parts. So your brain is covered by skull. Under the skull, you have the meninges. And inside the meninges, you see the fluid moving, that is CSF. And then under the CSF, there is another membrane, which is not here. And then you can see the brain tissue, which is amber color here. So this is the uppermost part of the brain is called cerebrum. And it is two half spheres, we call them cerebral hemisphere, right cerebral hemisphere and left cerebral hemisphere. Each sphere, cerebrum sphere is connected to other sphere through a bundle of axons, and that is called corpus callosum here. Under the corpus callosum, you have another part of the brain that is called diencephalon, this area. This diencephalon in the center, this area of the diencephalon is thalamus. This is hypothalamus, and around here and posteriorly it is epithalamus. Then from there, everything is brain stem. Brain stem is this midbrain, pons, and medulla oblongata. The pons and medulla oblongata, cerebellum is called hindbrain, and then your spinal cord is continuous. So this is the structure of the brain. In the brain, if you see the brain surface, it is never smooth. You see there is bumps, that is called gyrus, and fissure, that is called sulcus, groove, that is called sulcus. So human brain is big, and to fit them into our small cranium, it is squeezed, and that's why surface is like the foldings and enfoldings. So you have big surface area of the brain to fit a lot of larger size of cells, numbers in your small cranial cavity. That's why there is irregular surface and there is sulci and gyri plural. Okay. In the center of the brain, you have fluid. So if you go deep here, you will see the lateral ventricle, and here you will have third ventricle, here is fourth ventricle, and they all contain CSF fluid, and fluid from here comes outside and it also cover outside the brain. Okay, so this is the generalized idea of the brain. So CNS consists of brain and spinal cord, receives input from sensory neurons. So how our brain works? When you touch heart, that takes information from outside to your brain. So that receives, your brain central nervous system is receiving input from sensory neurons. Sensory neurons can be carrying any stimulus. It can be touch, pain, light, heat, anything. And then they process in the brain and then direct activity of motor neuron. So let's say if it is hot touch object you are touching, then our brain's motor neuron brings the impulse and then comes to the motor neuron, and from the motor neuron to your skeletal muscle, and you contract. So it directs motor activity. And there is in the middle, as I said, there are association neuron that integrate sensory and motor activity and perform complex function of the brain, which is learning, memory, higher mental function, judgment, calculation, emotion. Those all are the higher brain function. Okay. So CNS is composed of gray and white matter. If you take any brain and you unfold it, let's say you take a normal brain and then it is bumps and grooves. If you unfold them, then what happens? A normal brain size will be like average pillow size, large pillow size, not average, large pillow size. Okay. And if you give a frontal cut section through the bone, so let's say you cut and separate frontal portion of the brain and then see inside, what do you see? You see white matter in the inner part of the brain and the gray matter outer part, and some gray matter in the middle of the white part. So these gray matters are the area of the brain which is carrying neuron cell bodies and dendrites. So most of the cell bodies of the neuron in brain are towards the outer part of the brain, and that is called gray matter. The white matter are axons, and they are myelinated axon. Fats are whitish, and that's why they look white, and we call them white matter. Deep within the white matter, you have some group of nuclei, like diencephalon, and sometimes there is basal ganglia. Those are the special group of nuclei which is found only in the uh, human brain. Okay, those are called deep gray matters, but their name is nuclei. Okay, so adult brain weighs around 1.5 kg, contains 100 billion neurons, and receives 20% of blood flow to the body. So, like our brain, heart pumps like 5 liters of blood every minute. That means what is the 20% of 5? Around 500 ml our brain is getting. Okay. Now the development of the brain. So brain developed from the neural tube. So let's go evolution of human, how we produce human. So the female, here in the note bottom you can see, female makes egg, release on the 14th day of menstruation cycle, and then sperm comes. When egg and sperm fuse, we call them fertilization. Then what do we form? We form zygote, and then zygote has 3 layers: the upper layer, middle layer, and lower layer. Upper layer is called, most superficial layer is called ectoderm, middle layer is called mesoderm, and innermost layer is called endoderm. Okay, and the ectoderm slowly increase in size. Mesoderm forms muscle, bones, and other parts, cartilage. Endoderm forms epithelium and glands, and ectoderm forms neural tube. You see, so during 4th week, 3 swelling from a neural tube. So there is straight tube, but by the 3rd week, the ectoderm forms 3 tubes, and scientist has given his name. The most caudal, the rostral, the swellings towards the head, it's called prosencephalon, which is also called forebrain. Middle is called mesencephalon or midbrain, and the lower one is called rhomencephalon or hindbrain. By 5th week, by 5 secondary vesicles, by 5th week, there is the first proencephalon gives divided into 2, telencephalon and diencephalon. The second one remained 1, and third also divided into 2, metencephalon and myelencephalon. And now you compare the adult derivatives of the wall and cavities of the brain. So all your brain part, adult brain part, and the cavities inside the brain developed from this structure. So this telencephalon forms cerebral hemisphere, cerebrum. Diencephalon forms thalamus, hypothalamus, and which is not here, epithalamus. Mesencephalon forms midbrain, and rhombencephalon or hindbrain forms 2 swelling, which is metencephalon forms pons and cerebellum, and myelencephalon forms medulla oblongata. So hindbrain is pretty much pons, cerebellum, and medulla oblongata, and brain stem is midbrain, pons, and medulla oblongata, not the cerebellum. At the same time, cavity here is lateral ventricle, cavity in the 4th ventricle, in the diencephalon is 3rd ventricle, cavity in the midbrain is cerebral aqueduct, and cavity in the metencephalon is called 4th ventricle, or in the hindbrain is called 4th ventricle. That's how these all brain develops. Okay. Cerebrum is the largest part of the brain. 80% of mass is responsible for higher mental function. So all the outer cell bodies where there are gray matter, those areas are higher brain part. All the special function of the brain is there. Okay. Each hemisphere is called cerebrum. So brain cerebrum can be divided. Outer part of the brain, surface of the brain can be divided based on which bone cover them. So a frontal area of the brain is covered by frontal skull, frontal bone of the skull. Parietal is covered by parietal, temporal is covered by temporal, and occipital is covered by occipit. And each lobe is separated by deep fissure that is called sulcus. So frontal is separated from parietal by central sulcus. Temporal is separated from frontal and parietal by lateral sulcus. And then there is occipito parietal sulcus here. And two hemisphere is separated from each other by deep fissure that is called longitudinal fissure. And in the brain, you can see in front of central sulcus, this strip of gray matter that is precentral gyrus. Behind it, strip of gray matter, postcentral gyrus. Precentral gyrus is motor area of our brain. And postcentral gyrus is sensory area of our brain. Okay. We talked about this. We talked about gyrus, sulcus, all the lobes. And there is another lobe called fifth lobe of the brain that is called insula. So insula is also known as fifth lobe of the brain. What is that? So let's see, here is, this is, let's see, this is temporal lobe of the brain and this is frontal and parietal lobe of the brain, superior. So what happens, these two lobes slowly invaginate and buried inside, which you cannot see now here. And that inner part of these two lobes, which is buried inside, is called insula. And this lobe is associated with visceral memory and emotion. We're going to talk about that later. Okay. I think I'm going to stop here. And we will talk about that rest next time.
Professor?
Yeah.
So I know you said that the quiz is gonna consist of all these chapters that we went through, but are we gonna be able to complete chapter 8 and 9 before the quiz?
No.
No? Okay, so it's gonna be anything prior to today, right?
Yeah.
Okay.
Thank you, Professor.
Thank you.
Thank you.
Thank you.
Have a good day.
Thank you.